Right. So I'll just do a bit of an introduction. So welcome everyone to this evening's speaker series. We've got Dr. Parul on the call with us, and she's going to talk about her role as a technical operations manager at Illumina. And thank you for joining us. If you have any questions after the session, if you have any questions for Parul, you can email us at engage@welcomeconnectingscience.org or follow us on all of our social medias. And that is all from me this evening. So I'll hand over to our lovely guest speaker. And over to you.
Thanks. Thank you, Cindy. And thanks for having me. And thanks everyone for joining this evening. I'm really excited to share my journey, and I hope we can have some really interesting conversation and discussion, or I'm happy to take any questions as well. So I'm going to share my screen now. Hello. So thank you, Cindy, for the lovely introduction. I'm Dr. Parul Prabhat, and I'm going to talk to you about my journey. Moving on, I think I really wanted to start with this really a line which has stayed with me, a statement really, which is the journey is the reward. And this is something that I got from a book that I read, which is from Steve Jobs, and it really has stayed home with me. And I'll convince you all that actually it is the journey which is the reward, not really the results or what you've achieved, but it's how you've learned through the whole journey. And I think in this talk, how I've sort of structured is I'm going to talk about some of the growth, my journey in science, the research I've undertaken, and the different directions I've gone on to, because it's never always a single path that one takes. You sort of meander through different routes to get to your sort of aim, and I'll talk about a bit around that as well. So the agenda, double clicks. Agenda, I'm going to talk about my personal journey, education and highlights, professional journey, my role right now, and a little bit around genomic revolution, which I'm quite passionate about, and what does it mean to have a career in STEM. Before that, I think I really wanted to get our juices flowing and get your geography to test, really. I know it's a science talk, but I wanted the geography to start. And I wanted, I think Cindy is going to put a chat through, and if you can sort of guess which country I could be from these pictures.
We're not seeing the pictures yet.
Yeah, I know it's not moving.
Ooh.
Is it? Yeah.
Yeah, there we go. So where does it think?
What do people think? Wonder if you've been to this country. So yeah, let me and Cindy know. And yeah, please heads, let me know, Cindy, when I need to move.
Yeah, we'll just give a couple more minutes.
Yeah.
For a few responses.
Oh yeah.
Yeah. Just a couple more minutes. Awesome. So,
yeah.
Charity say India.
Ooh.
And a bit from Sri Lanka.
Oh, wow. Not bad. Not bad.
So where are you from?
I am what 82% of you said. I am from India, but I'm having some technical issues. It's not moving. Yes, I am from India, and precisely from Delhi, the capital city. And as you can see from the pictures, it's I was quite lucky to go live in a country which is as diverse in terms of culture, food, buildings, and people. So I guess I'm used to like living in a dynamic environment, which is sort of always changing, and that sort of preps you for what next for your future. So yeah, great. Everybody guessed it right. So yeah, I think you'll see a theme in my talk here is I like to quote some really famous people people I've read, followed, and and I would sort of tell everybody that I'm going to quiz you all at the end of the talk, that you remember the people I've quoted also. So yeah, please pay attention. So this one is another philosopher. He was an Indian philosopher, and it's a line which is sort of ethos of how I've lived, and how I sort of preach, or so my principles. It's dare to be free, dare to go as far as your thought leads, and dare to carry that out in your life. And so, as a young girl growing up in Delhi, I wanted to be a doctor. Didn't work out. It's quite competitive environment, so in the end, decided to do microbiology, molecular biology, go through the bachelor's, master's, and then I did a PhD. And now I am actually helping people, but not as a doctor, but as a scientist, and helping them in their genomic journey. And I'll talk a bit about it. So I guess this is where I would touch on the path or the direction. It's not necessary that you always do what you always had in mind as a child. You can always meander ways through or find alternates, which could also lead you to the same aim or goal. My father also has been a great influence in my life, and he's always said, if you set your mind to it, you will eventually get it. So and it sort of has stayed with me, and it's something I really passionately believe in. um so yeah, my journey. So I did my schooling in Delhi, and then I did my bachelor's in Delhi in microbiology. Then I moved on to I came to the UK to do my master's in applied biomolecular technology. I did that in Nottingham, and then I did my PhD at Imperial. And it has been such a lovely experience all through this whole educational journey, and I'll touch a bit upon it in my sort of next slide. Um, so yeah, the future depends on what we do in the present, and and why is that important? It's because if you follow your sort of passion, it's not necessary to think what next in the future, but you want to sort of uh, learn a lot from the present, and then find your own journey through that, and then decide what you do next. So like I said, I did my biology a degree, or sort of biology, in my school, and then I did my microbiology honors, which was kind of an alternate to medicine. So because I didn't get into medical school, I thought this would be a good way to learn the sciences and then see what next for me. And then I did my master's, which is what I said, in Nottingham. But through that master's, I got an experience to do an internship at Illumina, which was quite an enriching experience of how an industry works or sees sciences. And that really helped or shaped my thinking around how to pursue my career forward. So so I really encourage taking those experiences, taking those chances, and to understand what you want to do in the future through work experiences or internships, and they really can help you shape your future. Whilst doing my internship at Illumina, they offered me to work for them. So I did for 2 years. I worked with them as a research associate. While I was working at Illumina, it was very clear to me that if you want to sort of or sort of progress your career in sciences, you should doing a PhD is advisable, actually. And that was back in 2010, 2009. Things have really progressed since then, and it may not be necessary to do a PhD now to progress your career in sciences. But but what I sort of learned from my PhD is the experience and the wealth of knowledge you gain, and the confidence one gets from the PhD. So it may not necessarily be very financially rewarding sometimes to do a PhD, but it really prepares you for that career in science and that innovative streak in you, and to be very confident in your scientific knowledge. So, I mean, personally, my PhD experience was very, very enriching. And then since the PhD, I did maybe a few years as a postdoc just to finish things off, and sort of understand what academic life would mean, not as a student, but as an employee, which I did at University of Cambridge. But then since then, I've kind of been in industry working as a scientist or as a manager. And I'll touch a bit upon it as well as to how these two are different. But actually, that is almost like a good segue, Cindy, for our next slide, which is also a poll, I think, is what do you think are the basic differences between academic and industrial um, sort of scientific career?
So our poll is live. So which of these that Will has put on the screen, which do you think apply to academic science if you do your voting? Quite a few options. So we'll give a few more minutes for that as well.
Yeah, I think there's a giveaway here. I was supposed to do something here, which I didn't. Sorry, Cindy.
Oh, no.
It's an open book question. Never mind.
Oh, but we do have quite a mix of responses as well.
Oh, really?
Mm-hmm.
Oh, thank you, guys. You saved me. My big technological blunder has not been fully revealed.
Okay, so we will.
Yeah.
And that, yeah. And let me share the results. Can you see those?
Yeah.
So, fundamental research, self-motivated, accountability, publication, journal. It's quite a lot of consensus there and collaboration.
Yeah. Yeah. Great.
So what are the differences?
So yeah, what I think, sorry, it was not, I was not supposed to give you the names already in the table, but never mind. So industry, as I think you got it right, industry has a lot of dependency on obviously revenue. The benefits for the employee is great. It is a huge amount of team effort. It's not so much in isolation for an individual. It relies heavily on patenting your innovation, and there is a lot more accountability in the sense because of the money, the revenue at stake, and obviously it depends on kind of role you're in as well. But you got it right that industry on academic science is a very almost like a very defined differences between them. And in academic life, you really focus on publications or presenting in a conference. That's at the sort of the peak of your success would look like. It depends heavily on collaboration because in the sort of the day and age we live in, grants or the funding is not so it's not so I guess it's not so I mean, it doesn't come in such abundance. So you have to depend on a lot of scientific experiences from your colleagues or in your sort of research institute, so that you can leverage on each other's expertise and a bit around their funding, so that you can collaborate and bring out a very relevant research. And like I said, a grant funding is at the pinnacle of it all, right? If your research fully depends on it, so you kind of have to really work hard to make it relevant for those grant funders to give you the money to do the research. In industry, obviously it exists, but there is a bit more money, especially in the US. The money is a bit more for the industrial research than a sort of academic. And obviously academic research is fundamental sciences, it's the basic sciences, and I personally think that should be the core of an academic research, because that's the foundation for industrial research to go on to. So if you don't have basic sciences, it's not going to lead anywhere, the industrial research. So they go hand in hand. But with my little experience I've had, there still is a lot to be done around this collaboration between industry and academia. It still is not as good as one would anticipate because of those blockers. Academic sciences needs journals, industry needs patents. So they don't want to always, academic sciences don't want to make their research so restrictive, right? They want everybody to be they want their research to be accessible. So that's where I think the big sort of contradiction comes between the two. And obviously academic sciences, it's very self-motivated and it's self-driven. So there is a lot around individual achievement and individual how an individual is performing in their research. So that can sometimes be sort of demoralizing, because as personally you would have put your 100%, but not everybody believes in that your 100% was good enough. So it does make a huge difference. So yeah, I think good that we kind of got you guys. I think the audience is understanding the nuances between the academic and industrial scientific career. I'm really glad. And I guess I want to also show an image. It may not be exactly how it is. It's a bit stereotypical, but the one which is highlighted in blue is most likely a typical academic lab could look like, and the one highlighted in white is how an industrial lab would look like. Not all of them, but especially a high throughput genomic lab. So this image is from the lab I work in, and it's, as you can see the differences, it's highly automated. It has very, very efficient Six Sigma lean type, which is called, it's a way of manufacturing jargon, but it's very, very structured, very organized compared to an academic lab would look like. But yeah, just to highlight.
Yeah, quite the difference between the labs.
Yeah,
My goodness.
yeah. But somehow, as they say, great science happens sometimes in chaos, and that's what most academic labs want to live by. They want to show the chaos to show that they are doing great science. Moving on, I thought it'll be good segue to talk about my insights from my academic career. So this is a bit of sort of excerpt from my PhD. And like I said, my PhD, I did 3 years, and every single day of my PhD, I thoroughly enjoyed. But that is a lot of credit needs to be given to my supervisor, my lab, the environment, the people I was doing PhD with. It was just brilliant. So I think Cindy must have shared some of the papers that I published in my PhD, but the highlights from my PhD were I developed a novel method in looking in microscopy, which was extremely high resolution, which was looking at each and every single E. coli cell. I don't know if you've read in your biology, but E. coli it's a tiny microorganism which is one of the most studied organism in the sciences. Every lab would use E. coli if for any basics research. And each and every cell, if you see in the image, is actually an E. coli cell, and a single dot that you see inside it is the protein complex that I've shown here. So these protein complexes, what I'm visualizing in the E. coli. So it was quite eye-opening for me. A lot of hours in a dark room were spent creating these images, but it was very rewarding. And so in that PhD, I did a lot of different biochemistry, microscopy. I did not just the sciences part of it. I did collaboration. There was a lot of highly advanced mathematical programming to look at these dots and to represent them into a quantitative scenario as well. So it was not just qualitative. And the soft skills, I guess, what I learned from my PhD were innovation and understanding how to create new ideas and how to actually implement new ideas. Troubleshooting every single day in a scientific lab is all about troubleshooting. You always have from a smallest of thing like a buffer you're not able to make, or the constant, or you're not able to find a solution to really highly complex problems, which in the end you might have to lean on to somebody else's expertise. So there is troubleshooting at a daily basis. Obviously, accountability, initiative, leadership, supervision, proactivity, scientific communication, and illustration. So I mean, all this, as you can see, is all I made it for my PhD. So you actually not just learn how to talk about your science. You also have to present it in a nice way, because when you go in conferences, not everybody is thick into your work, right? So you kind of have to make it understandable by the wider audience. And like I said, collaboration. So it was really a very enriching, whole-rounded experience. It was not just being geeking on a nice scientific paper. It was about convincing people of how you have done all this work, and how this is relevant, and how this is correct. Um, so yeah, I think that is what was take home from my PhD, is a lot of scientific skills, but a lot many soft skills, which are relevant in any field beyond scientific research. And then after my PhD, I moved on um, to doing a postdoc in the same lab, and also then briefly in my postdoc in my PhD lab. But then I moved on to do a postdoc at University of Cambridge. Uh, and there I took the same sort of ethos around microscopy, but then I moved on to a different organism, which is Baker's yeast. It's a model organism that this lab was working on. There I looked at, again, microscopy, enzymes, how they sort of localize in a cell. And it was, again, very enriching experience. And exactly the same skills, I moved on and took it, learned and developed upon it. And this was also the time when I had my son. So it was a very interesting career move for me, or career change. I was moving from having a very scientific career to taking a break in my career. And that was the time when I actually got time to think what next for me. And that's when I kind of almost had decided that I want to move on from academia to industry, because I was not able to see the gains for my career in the academic life. And I thought industry would be a great way to evolve my sort of scientific career, and hopefully do the things that I always wanted to do potentially as a medic. Now I could do as a researcher, and change people's lives and patients' lives, and help in human health, really. Because all this so far I've done is basic sciences. I was not really seeing any opportunity to do work which could be relevant to the wider to human health, really. So I guess this was a very interesting time where I actually I was off for a maternity leave for a year, and that's when I sort of thought as to what does a scientific career look like. And after having done a PhD, a postdoc, am I stuck in doing a postdoc? Will I get other opportunities? How do I look for it? And then actually I sat and sort of wrote down the skills that I have, and all the sort of qualities I've developed, and I saw that I could take so many different directions my career could move to: consultancy, finance, quality, marketing, entrepreneurship, project management, research, and data science. For me, I took research as my focus, and then I moved on to industry. But it's just highlighting how you think so when you're doing a PhD or a master's, you think you will be just doing sciences of all your life. And if you feel stuck, just think openly of all the sort of resources you've developed over these studies, and you realize it's very transferable skills that you've gained while studying sciences. So that's what I did. I started looking for jobs in industry, and like I said, I highlighted there were such relevant transferable skills that I could move to a career. I could make the career move to industry. So the expertise in scientific research, innovation, curiosity to learn, understanding scientific journals, communication skills, leadership, supervision, project management, and collaboration. All these skills were very, very important and key to getting success at interviews and getting the job that I have in the past sort of 8 years. And it's very important to highlight, actually, this is a very key year. And when I was putting these slides together, I realized this is the year when I would break even almost. So I would have spent 50% of my career studying in academia or being in academia. And now I would have spent the same amount of time working in industry. So it's quite an interesting time for me that this is it. I'm not going back to studying anymore. It's industry is my choice for career, and I'm so pleased for it, that I made that decision. So yeah, I mean, I guess I've spoken a lot about skills and things like that, but it's not just that. I really enjoy doing things outside of work. I found this off the internet, and I thought it was very interesting. And it's exactly like that. I have a split personality almost. There's a scientific part of me, and there's this really social part of me. And I do count. Don't count the days, make the days count. I really believe in it. So yeah, just quickly jumping on to that. Ooh, I didn't want that. No, let's do that. So I really enjoy doing things outside of work. I spend a lot of time traveling with my family. I love cooking, entertaining friends, eating out with my family. I love shopping, dancing, folk dancing. So yeah, these are just some snippets. And having two kids, they keep me on my toes as well. So yeah, I spend a lot of time, having fun with them. So yeah, moving on from my hobbies and outside of work, I want to move on to sort of industry. And what do I do in my current role, and what does genomic mean, and the future of it. So before that, I wanted to gauge how many of you have sort of worked in the lab, would have seen some of these equipments in the lab. So please put in the chat if you recognize any of these. I've numbered them, and then of course I can, just to get ourselves in the scientific mode, really. So yeah, we'll give you some few minutes, I think, on it.
Yeah, so if everyone can add their responses in the chat box. If you can't access the chat box, can you add in the Q&A section? I think I've enabled it. But does anyone recognize any of these pieces of equipment? Have you used any of them, or know what they're called? What they might be used for?
Sorry, some of them I've put in some tricky ones you may not have seen in a school lab, possibly not. And if you have, good for you. I mean, you work in some really fancy labs. But yeah, I thought
they do look very interesting, these items. So we've got Bunsen burner for number 3.
Yeah.
Got mass balance for number 4.
Yes.
Number 10, is that a centrifuge?
Very good. Yes. Tabletop centrifuge is what we call it.
Cool. Number 7, a Petri dish.
Yeah.
Awesome.
Any guesses for the big ones? These 15 or 12? 14 is a bit obvious, I would have thought.
Hopefully so. Yeah. But it looks a bit different to a lot of the ones I've seen in a lab. Bit more specialist. Is that because it's hooked up to a monitor?
Yes, exactly. Yeah, bigger monitors and things like that. Yeah,
we've
definitely.
got microscopes. So yeah,
very good.
I think 13 looks really weird to me. Oh, 14, is that electronic microscopes? 15, mass spectrometer?
No, actually, but very good, very good guess. 15 actually is a personal favorite. I know, odd. This is it's a few million pounds. Oof. It's an omics microscope. And actually, all 3 things are part of the microscope. The microscope actually sits inside this. So you have to sort of walk in and open it. And it's really one of the fanciest microscopes you could buy. And very fortunate, I worked on it in my PhD. So I wanted to put an image of it, and I was so impressed. It does do really high-tech microscopy for really tiny microorganisms, viruses.
Wow, that sounds awesome.
Human cells, human tissues. I think there are 2 in the UK. Ooh. Only 2.
So
yeah.
someone's also taken a guess at what 13 is. Is it an ultracentrifuge?
Very good. Correct.
Oh my goodness, that's a
It is.
centrifuge?
It is for basically ultracentrifuges is used to sort of extract protein. So basically, what you do is you really sort of, what do you call it, degenerate the cells, so that can be used to purify proteins. So the number 12, I don't know if anybody's not guessed, is actually called a protein purification machine. So
Ooh.
I wanted to put the two together. So you basically spit cells down in this really crazy speed, and then use the solvents to purify your proteins from it. So yeah, it's one of the I don't know, like a godsend for a biochemistry lab. And 9 is actually a gel loading machine. So you run your proteins and DNA, most likely proteins in it, and to see if you've got impurities or not in your protein.
So would
So
that be your PCR?
it's not PCR. It's called a gel dock station, or gel running machine, and you sort of run the gels, and you sort of put your protein a solution in it, and then it runs through electronically, sort of, on the charge basis. um, because as you see, there's negative to positive, and then your protein sort of flows through, and if depending on the size of the protein, it would sort of run till a certain point, and then you have sort of molecular weights running on the side, and you can define what size it is. So yeah, it's quite one of the very important machines if you're doing DNA work, protein work, to understand if there are impurities. So yeah, great. I think you guys guessed it. Um, so moving on, like, oddly, none of these would be present in a genomics lab. But what I wanted to give a flavor was labs are of many different types, and different machines are present in labs depending on the expertise of the lab, right? So this is what my PhD lab would have looked like. But now industrial labs are a bit different. Um, so I think moving on to sort of industry and what my current role is, I wanted to highlight um, what does Illumina Lab Services, or Illumina, uh, do, or what my lab does actually. And what we do is we process clinical samples. So you can imagine, it's how it looks like is a patient would go to a doctor. If you have a certain type of disease, a rare disease or a cancer, now in the UK, a doctor can prescribe, like any blood test, like a lipid test or a diabetes test, they can prescribe a whole genome sequencing test for a patient. And once that is decided that this patient will get that, the sample is collected, which is blood or sometimes tissue. All that is taken to a central processing hub, which is run by NHS, and they extract DNA from those samples, and they send it to Illumina Lab Services in a format of a 9612 plate. Then comes Illumina Lab Services, which is where I work. We make these DNA samples ready for processing, and then we do something called DNA quantification. We prepare it to be processed for the next stages, and the crux of it is we send sort of data to certain data processing labs, and then they create a report for a clinician to then advise the patient what is their next what is the next treatment for them, or what is next for them in their journey. Right now, there are 21 diseases that they can get whole genome sequencing for um, in NHS. And NHS is the only place in the world right now which provides free whole genome sequencing to their citizens, which is, I think, quite an incredible achievement for NHS. We process around 30,000 samples a year. We are a clinically accredited lab. It's called an ISO 15819. Because of that, we have to follow very strict quality guidelines. and everything has to be done and documented in a certain quality QMS procedure. And our turnaround time, which is from getting a sample to delivering that data, is 21 to 28 days, depending on the disease type. This is what my lab uh, sort of fits into the whole clinical journey of a patient. Um, and just to show an example of how the lab looks like, you've seen the image before. This is an image from the lab. The image on your right is an image from from the web, but our lab also looks like that. And the machines you see in the image are something called sequencing machines, which is where we put our DNA to get a readout.
Which they can run a whole, how long does it take them to run a sample?
So yeah, it takes 3 days for us to process a sample.
That's all. I think it took 13 years to do the first human genome. So now
Exactly.
it's 3 days.
3 days. And that's
Oh,
not
my
just one
word.
genome. We sequence 20 genomes at a time, or 8. So we get in 3 days, 3 to 4 days, we could get on a good day though, Cindy, you don't always have a good day in the lab. So therefore we have a huge margin. We have 21 to 28 days to correct if something goes wrong in the process. But theoretically, everything going well, we can deliver in 5 days.
That is
Um,
amazing. And
yeah.
that many samples a month. Oh, wow.
Yeah. I mean, so 6,000 exactly. So I guess to put in perspective, we have only 16 lab members, lab team. Obviously, we have many more aspects of the team. So there's lab, there's bioinformatics, and software. So actually, you would think a scientific lab would just need lab people. Not really. A genomic lab needs actually software developers, bioinformaticians, laboratory, quality, finance, supply chain, you name it. It's a whole industry in itself, really. And in the lab, we have 16 lab members, we have 6,000 samples, and we have 12 liquid handlers and 14 sequencing instruments. And each of that instrument is £1 million, by the way. So, it's a lot of investment in a genomic lab. What does my role look like, and what do I do on a daily basis? Again, you'll see some common themes here: troubleshooting, team building, team development, process improvements, goals, activities. So there is a bit of a running theme. So from academia, from industry, and now to this role, which is a bit more managerial, more leadership. That it's the same theme. I've used those skills and just developed and built on the career. So again, I'm the subject matter expert in the lab for the technology development, for technology transfers, for continual improvement and innovation. I help in that as well. There is a separate team which takes on these projects, and then I sort of help them in their core team meetings or in any other troubleshooting that we may face on daily basis. So there's things which you do on a run the mill basis, and there are some things which are sort of goal driven, which are more long term. So that's quite an interesting role, actually. A lot of team building. So I don't line manage, but I project manage many sort of colleagues and people in the lab. There's a lot of focus on celebrating successes, acknowledging teams, a lot of cross functional networking. We do lab tours. I focus on my career development, but I also help others in the team go to conferences, do external courses and training. And this actually, the STEM future, is also because I came into Illumina and met Cindy, and this is how it all stemmed from, really. So I think it's not just helping in a daily day to day running of the lab, but there's also a lot more into personal development for myself and for the team. A lot around process management, QMS, bioinformatic management, software management, and of course, like I said, everything here is goal driven. So we always, if we have ideas, we pitch it as a goal and see if there is any sort of appetite for it to go forward as a goal, so that we can track progress. It's not just written in my notebook, right? It is measured. It is discussed. If I have issues with it, we have, um, meetings to discuss progress and roadblockers. So there is a lot of focus on goal driven work. But in this, I wanted to, I think, highlight and ask a question. Now that you've learned a little bit about genomics or gene sequencing, um, what do you think a genomic data, when I say a genome, how much data would that produce? Can anybody guess? I think, Cindy, do you want to run another?
Yeah, so we've got a poll live. It's in gigabytes, I think. So how big do you think the data output for one genome is? Ooh, got a few. It's interesting. I can't remember what the answer was, to be honest. Ooh. So we'll just give that a couple more minutes and see what people think.
Time-wise, how are we doing?
5, 10 minutes, and then we'll go to Q&A.
Okay, great.
Okay.
Should I reveal? Dun dun.
I'm going to end the poll there and share the results. So the majority say 128.
Wow.
Or 512.
Brilliant. We have a very clever audience. I don't think so. I need to tell them anything about genomics now.
So how much is it? How big?
Very good. Yeah, very bang on. A 128 gigabyte iPhone. But that's one genome, and we are doing 30,000. So yeah, you're very correct. And to put in perspective, actually, this was a very powerful slide which I found, and you can read the paper, is the complexity of human genome. And if you can see how it compares or fares against all the other technological advancements, it doesn't touch it. We have 3.3 billion base pairs which we have to read in a human genome, a single human genome. And compared to an iPhone, a Hubble, or the Mars Curiosity rover, or all the cool missiles or airline industry, they don't even come close to it. There's 44 million codes in that technology. So actually, human genome is very, very complex. So I think we need to give credit to all the work and research that's gone over the decade in making it simplified so that we can read it and understand it and decipher the complexities of it. So I just wanted to sort of highlight that. And quickly, as I highlighted, the skills I learned in my PhDs, my postdocs, in my master's internship, they're still so relevant and still so helpful in my day-to-day work right now. And it gives me confidence to do my job even better because of all these skills that I've honed over the years. So I just wanted to highlight that. And quickly, about Illumina, very, very quickly. If you've heard of it, I think we'll skip this one, I think, Cindy, or should we do it? Skip this one, right?
Yeah.
Yeah. So Illumina is a very very interesting company which has really revolutionized how human health is perceived now. And their motto is to improve human health by unlocking the power of the genome. And that's what they do. And why is this so important? Human genome is actually packed into a small cotton wool which is in the nucleus of a cell. And when you really open your lens on it and put your microscope in it, it goes from a chromosome, unfolds itself into this really tiny complex structure, but at the core of it is this double helix, which is what we are studying on a daily basis in our lab. And there is so much useful information in that double helix that then manifests itself into all these changes in the cell, that it is just incredible amount of information one gets from that, reading the human double helix. And what you're reading in your double helix is these adenine, cytosine, thymine, and guanine bases. But actually, it's not just restricted to it. There is much more complexity to this, ACTG, there's much more chemical changes to them, but at the core of it is what we are studying in a sequence. And how Illumina helps in that is what they've developed is the whole suite of things. It's a whole package. You can do you get your DNA, you make it next generation sequencing ready. So you add uh, adapters, primers, you put in this amazing instrument, and then what you get out of it is this sequence of information of 3.3 billion base pairs. And that's how bioinformatics has evolved in understanding those bases. I think let's do this one quickly, is where do you think, with these pictures also, where would you think genomics could impact, in what markets? Quickly, if you can put some thoughts there, and then I'll give you the answers. And it'll be quite powerful as to where all genomics can actually um, impact.
So yeah, if you want to just put in the chat box, everyone, so these pictures, like what I said, they are hints. And what markets do you think genomics impacts? So can you give us an example?
Yeah, I could. Yeah. So if we look here, that's I think there's a hint. There's a lot of work where genomics has changed the whole reproductive health world, where where I don't know if you've heard of it, the IVF, the in vitro fertilization, and how it has changed and helped many families have children when they could not have had children. So this reproductive health is a huge market where genomics has helped, and also helped families where they found out that there might be some abnormalities with their fetus, and then how to correct it, or at least identify it before, so that they're prepared for it. It's helped there. It's
Awesome.
I mean,
And we've got just a quick question, just going back a minute.
yeah,
What were the full bases of DNA again?
it's adenine, cytosine, thymine, and guanine.
Lovely. And then we've got some guesses. So does genomics impact pharmaceuticals?
Yes.
Does it impact maybe someone says the food industry, to see how we can genetically modify the food to make it better for us?
Correct. Yeah.
Awesome. And what are the others? So you've just revealed some names.
Revealed, yes. So there's obviously reproductive health that I touched on in this image. And there is rare diseases, which is what we actively study in our lab. There is oncology, which is cancer. Cancer genomics has totally changed the way cancer treatment is done. Research, generally, basic sciences, just to sequence small organisms, big organisms, plants, general research. Infectious diseases, you all know, COVID sequencing was all about Illumina sequencing. So the whole surveillance, everything was because of sequencing. We could catch, I mean, the vaccine was created because we could sequence the virus so quickly and then get the vaccine into the market. Drug discovery, like one of you mentioned, proteomics is another one, which is really cool. Is basically you look at proteins are present in your multivitamins, in your supplements, and how you could study those proteins and change them and be useful for your health, general health, in drugs and things like that. And of course, agriculture. Yeah, exactly, huge market. So I think I'm almost concluding here now, and I'm sure many of you would have read it. Winnie the Pooh, and it has always been such a lovely image for me. And the saying, "It never hurts to keep looking for sunshine," is so dear to me, and that's why I want to finish by saying that the path, the priority, and passion are the key words I would take home from my slides. Stick to the path, or have a path in your sort of mind, that whatever it is, education, training, apprenticeship, mentors. So have all this sort of sketched out in your sort of plan for future. At least know the priorities, where your priorities lie. And at the core of it all is the passion, or follow your passion, whether it's sciences or whether it's a particular stream in sciences. And also passion for sort of teams, people, is at least the core for me in my career. And I wanted to highlight these 3 Ps, which I kind of live by, and really sort of, if anybody asks for advice, I would give these, keep those 3 clear in your mind. And some sort of top tips is, again, it's just moving on from the previous slide. It's just have some constant mentor or mentors. I have few. I call it my personal board of directors. And I go to them. If I have any questions, I'd go to them, ask opinions, career choices, personal choices. Yeah, I would really recommend to have. And to highlight that STEM is such an amazing sciences, or sort of path to follow, because it's very multidimensional. As I highlighted, you can pursue many different careers even after studying sciences. It doesn't need to be research. It can be many other different things. And like I said, I think I want you to, do you recognize any of these inspirational people and characters from my talk? There was a general theme? I quoted them, and I thought I'll give them a little brief mention in my end credits if you recognize them. This is, of course, Steve Jobs. This is Winnie the Pooh. This is Mahatma Gandhi. This is Swami Vivekananda, the Indian philosopher. This is APJ Abdul Kalam. I've read a lot of their books. He changed India's space research. So these are some of the people I really have got inspiration from, or read about, or enjoyed reading. Winnie the Pooh, it's just a lovely book to read. So yeah, I think I would probably end my talk with that. And thank you so much for listening. I hope you enjoyed it. I really enjoyed it. And it was such a lovely look back for me to go through and put these slides together. It was really amazing experience. Thank you.
Thank you very much. So I'll give you a round of applause for everyone.
Thank you.
And we've got a thank you from the audience as well.
Thanks.
I think it's just amazing how you've gone through a few different, like you said, academic and then industry, and it's been quite varied. Is there any part that you have particularly enjoyed the most, or is it now?
Like I think I really would mention that my PhD was one of my, I think, the best decisions, because I was contemplating between doing an MBA or a PhD. And I am so glad that I did a PhD instead of an MBA.
And what's an MBA?
So yeah, a management degree. So management in business administration is what I thought I would do after my master's. And that was always my goal, basically. Maybe move into management of medical research or management of that. But then to do a PhD really changed how I pursued my career. So I think my PhD and now my current role, it's been really amazing experiences in my career, I think, so far.
Fantastic. So do we have any last questions for our scientists today? If you have any, please can you put them in the chat box or the Q&A? And we'll give a few minutes for that. And I am just going to share screen.
Yeah. Can you get the controls? Should I stop sharing, you think?
Yeah, please. Thank you. How long did it take you to move to industry from academia?
Actually, not very long. So I moved. So when I was on my maternity leave, I applied for my for a role in industry in my previous company. So yeah, it was a very seamless transition, actually. But like I said, I think it's the way you present yourself. So I would really give an advice saying that have skills which are not very specific to your work. Have a very transferable skill set that you show in your CV. Everybody has it. It's the way you present it. That's what matters. And I think I learned during my PhD as well, that how to present your skills and in a transferable way, and therefore the transition was not very difficult actually for me.
Oh, that's good. Yeah, because a lot of the skills that students are developing at the moment in all of your studies, you will be applying to what you're doing now, and you can add that to your CV on how you are transferable, just like Parul says. Just a quick reminder as well about logging your activity, so making sure that you have noted that you've attended, so thank you very much. And just that our next session is next week by Ash, and he'll talk to us about endocrinology and diabetes. And you log your hours on, I believe, the Youth STEM Award Hub. Oh, why is that not going back? Where you would generally log all your other activities for this. But if you have any questions or concerns on that, you can email me directly on, it should be the website. But I will add in our email on the chat box that if you have any questions on doing that further, I can send you more on logging it, or any further questions for our lovely guest today. Thank you very much for that presentation. I really enjoyed having a sneak peek into your world.
Thank you for having me. Really.
It's quite different. Definitely. Yeah. And I don't know, it was that easy to hopefully switch between academic and industry, and the differences between science in the two worlds. Quite interesting as well. Thank you so much.
Thanks a lot. Thank you.
And thank you everyone for attending. I hope you enjoy your evenings. I think there's some fireworks going on tonight. I know they're on campus.
Yeah.
I hope to see you all soon.
Yeah. Bye-bye. Thank you so much. Thanks.
Have a good evening.
Bye. Bye-bye. Bye-bye.